Hello everybody, Mrs. Horn here. We're kicking off our last unit in AP this year with the animal homeostasis unit. We're going to start talking about the digestive system. We're going to touch on the difference between intracellular and extracellular digestion and then move right into human digestive system and accessory organs and part of the alimentary canal and different enzymes that are going to digest different types of organic compounds. So here we go. There are four main stages in the processing of food. Food has to be ingested, then it will be digested, then it will be absorbed into the blood, and then it will be eliminated. Unused material will be eliminated. There's a big difference between an organism that carries on digestion through intracellular digestion versus extracellular. Now, intracellular digestion is when food is ingested through phagocytosis or pinocytosis. Normally, this is in something that's really small, microscopic, maybe even only one cell big. A bacteria cell would do this, an amoeba, paramecium could do this. And when they ingest the food gets encapsulated in a vacuole, and then that vacuole will fuse with a lysosome, and we know that lysosomes have digestive enzymes in there, and a lysosome would help break up all of the food so that it becomes a usable size for that organism. Now, extracellular digestion is when the digestion occurs outside of the cell. This usually happens when an organism eats something that's bigger than itself. It may have a single opening system. We call this a gastrovascular cavity. A hydra is a good example of something that carries on digestion like this. There's a picture in your textbook if you want to take a look at it. Hydra is a pretty cool thing. It's, again, microscopic, really small, lives in fresh water, and it looks like Medusa. It has a bunch of tentacles on the top of it, and that's how it will paralyze and bring its prey in. Extracellular digestion could also be for a system that has two openings, and we call this the alimentary canal. And food will move in one direction, so it moves in through the mouth and goes through the whole system, and then any undigested material would exit through the anus. So we have an extracellular digestive system. In mammals, our digestive system consists of an alimentary canal and a bunch of accessory organs, and we're going to start talking about the importance of all those accessory organs. Starting at the top of the alimentary canal, we have the salivary glands. Salivary glands secrete salivary amylase. This is a chemical that will digest only carbohydrates. So the only chemical digestion that takes place in the mouth is that of carbohydrates. The pancreas is another accessory organ that produces something called pancreatic juice. It's going to secrete this substance into the small intestine. Pancreas is also part of the endocrine system, and we'll talk about that later, so it does have a dual purpose. The liver is an organ that makes bile, and again, it has other functions, but in the digestive system, it makes bile and then releases it into the gallbladder, and the gallbladder is going to hold on to it for a while and release it into the small intestine. Now, bile is a substance that will help emulsify fat. So lipids that you consume, they're going to get broken into smaller pieces through this bile. And then we have a picture at the bottom. Nothing is labeled on here, so I'm just going to tell you what everything is. This structure here that looks kind of red, that's your liver. Liver is very large. That will make the bile, and it will deposit the bile into the gallbladder, which is this little green, looks like a balloon, and it actually does look green when we dissect the cat. You'll see it looks kind of green. This yellow structure here, that's the pancreas. This is the stomach, which is not an accessory organ, but it's nice to see it in a diagram so that you see where it is in relation to other things. And then you can also notice that the pancreas has a little tube connecting from the pancreas into this larger tube, and the gallbladder is also connected to the same spot. That's where the bile and the pancreatic juice are going to enter the small intestine. For the alimentary canal, so this is where food will enter through the mouth, going to enter through the oral cavity, and then as you swallow, it's going to travel through the pharynx. That is what connects the mouth to the esophagus. Once food enters the esophagus, the esophagus will deliver the food through the stomach, and the way in which it moves is an action called peristalsis. And peristalsis isn't that the food just simply drops down into your stomach, but the food is pushed through the muscle of the esophagus, and it's squeezed and squeezed and squeezed, and finally the food gets down into the stomach. So it's not an instantaneous drop into the stomach, but the esophagus itself is a smooth muscle that is an involuntary movement that will move that food. It will squeeze that food down into your stomach for you. Once the food gets into the stomach, the food will be stored there, some of it, so that you don't have to eat all the time. The stomach will secrete a very acidic gastric juice. It's a very low pH of about 2, and this will help chemically digest proteins. And this is also made of a smooth muscle. It's an involuntary movement. You don't have to think about the stomach breaking apart food. And the stomach actually is another area of mechanical digestion. So both chemical and chemical digestion are taking place in the stomach. Stomach, which is a muscle, a very strong muscle, will move itself and squeeze itself and push itself, and as it does it, it's breaking apart food, just like your teeth break apart food when you chew on them. That's mechanical digestion. The chemical digestion is when you involve the gastric juice and the enzymes and the acids that destroy or break up the chemical formation of the organic compounds. From the stomach, food moves into the small intestine, and this is where most of the chemical digestion happens. So here we're going to digest proteins, carbohydrates, lipids, nucleic acids, which I don't have written on there, but they will be digested there. And then this is where they're getting into their smallest components, those really small monomers, those simple sugars, those amino acids, the fatty acids and glycerol molecules. And once they're that small, that's where absorption takes place. So from the walls of the small intestine, those monomers, those really small organic molecules, are going to be absorbed through the walls of the small intestine and into the bloodstream, and then the blood can circulate all of these nutrients throughout the body into every cell. And the structure of the small intestine that where the molecules are absorbed through is called the villi. I've got a picture of that here in just a minute. Now, the large intestine, if anything gets to the large intestine, it's because your body didn't really want to use it. So the undigested material is going to end up in the large intestine along with water. Now, the main purpose of the large intestine is to reabsorb water. So depending on how much water your body needs, if you need more water, it's going to absorb more water from the fecal material. If you don't need a lot of water, because maybe you're drinking a lot of water, it might leave some water in the fecal material. The large intestine could malfunction, and if it's not reabsorbing the correct amount of water, it could lead to diarrhea if it's leaving too much water in the large intestine, or it could lead to constipation if it's reabsorbing too much water. The picture I have here shows everything I was just talking about, and nothing is labeled, so let me quickly walk you through what everything is. So up here in the mouth we have the salivary glands. This is the esophagus, so the food travels through, and the food is squeezed through with that smooth muscle. It will pass through a strong sphincter muscle here and go into the stomach. The stomach will then chemically digest some proteins and mechanically digest food. The food will then pass through another sphincter muscle and into the small intestine. The small intestine is about 21 feet in length, but called the small intestine because of its small diameter. Lots of chemical digestion is happening here. Carbs, proteins, lipids, nucleic acid, everything you can imagine is being chemically digested here, and then absorbed. The nutrients are absorbed into the blood, The large intestine is the structure on the outside of that, large in that it's very thick diameter, wide diameter, but much smaller in length, about 5 to 6 feet compared to the small intestine. On the next page of your packet is a chart that gives you specific details of names of organic molecules, where they are digested, and even the name of the enzyme that's doing the digesting. So the chart that you have on your page is oriented in a landscape format, but the one I have on the screen is a portrait picture or portrait setup. So make sure you're writing everything in the right box. In the mouth, the polysaccharides, which are very big and complex carbohydrates, we can also call them starch, they are chemically digested by amylase into smaller polypeptides. Maltose would be a good example. Nothing else is chemically digested in the mouth other than carbohydrates. In the stomach, the only thing that's chemically digested there are proteins, so really big polypeptides. Pepsin is the name of the enzyme that is produced in the stomach, and they're broken down into much smaller polypeptides. So they're not in their smallest components yet. That's not going to happen until the small intestine. And once we get to the small intestine, which we have a lot of chemical digestion happening here, this is a little overwhelming to look at all at once, so I'm going to only show you part of it. In the lumen of the small intestine, we're digesting everything. So if you look at the polysaccharides, so our big carbohydrates, we're using pancreatic amylase. It's breaking it down into disaccharides, which are carbohydrates, two-rung carbohydrates. The proteins, which were the small polypeptides up in the stomach, we're using trypsin here in this part of the small intestine, and we're breaking them down into amino acids. The nucleic acids, the DNA, the RNA that makes up all living things, that's in everything that you eat. We're using a nuclease. It's breaking down into nucleotides. And then our fats. Our fats, we haven't broken them down anywhere yet. We're going to use that bile that was made in the liver. It's going to be broken down into some fat droplets. And then there are also some lipases in the lumen of the small intestine, which are going to break it down into the simplest form, the glycerol and the fatty acids. And then in the last section here, in the villi of the small intestine, this is where we're finally going to get all of our very small monomers. So we have our monosaccharides, our smallest particles of the carbohydrates. That's our glucose and our simple sugars. We're finally seeing some amino acids from our proteins. And for the DNA and the RNA, now we are seeing nucleotides. The components of the nucleotides. So we have nitrogenous bases, the sugars, and the phosphates. And the fats were already completely digested in the lumen of the small intestine. I went through the information really fast, so if you need to go back and get anything in the charts, please make sure you pause and get that completed. And the last picture I have for your packets, which I think my pages went out of order, but this should be page 7 in your packet. This is a zoomed in version of the small intestine. So this tube is a piece of the small intestine, and if you were to open it up and roll it out and look really closely with a very powerful microscope, you would see these little finger-like structures, and those finger-like structures are called the villi. Now I've got a zoomed in version of one finger-like structure, and what happens here is that the food and the nutrients from inside of the small intestine will diffuse through that wall, and it will get picked up by the blood, and the blood will then carry the nutrients to different parts of the body. But at this point, the villi are so important because they're increasing the surface area necessary to diffuse the amount of nutrients at a very efficient rate. If all these villi were not here, enough nutrients wouldn't be diffusing fast enough for a very big and complex human to have enough nutrients or other requirements that they need throughout their day in all billions of cells that they have. So the villi are really important to help increase the surface area so that those glucose molecules and amino acids and nucleotides can diffuse into the blood at a really fast rate. So this gets us through the digestive system, and I hope this helps.